Okay, so we're getting into the cells now. We're going to be living in the world of cells for several weeks. Chapters 3, 4, and 5 are all about cells and cellular processes. And so we're going to be here for quite a while. It's a very important concept in biology. All living things are made of cells. The cell is the basic unit of life, and so we need to become friends with cells. The story of the discovery of cells is mostly a Northern European story. The science that allowed the discovery of the cell is based on the microscope, and the microscopes were developed in Northern Europe. And so a lot of the first people to study cells were either English, Dutch, or German. So Robert Hooke actually did tons of things in his life. If you've ever heard the phrase Renaissance man, as in somebody who can do everything, that's Robert Hooke. Robert Hooke was a theologian. He contributed much to the writings of the church at the time. He was also a scientist of many, many different disciplines. He did some geology, he did some physics, and he did some biology. His physics was grinding lenses. He did some early optical work. He helped with a lot of the early science that has become my glasses, was Robert Hooke. And as a consequence of grinding lenses and being able to magnify images, he was also the first person to see a cell. And so using a pair of lenses that he could get at the right focal distance from an object underneath, he magnified cork. And, from like a wine bottle or something like that he had sliced real thin. And he saw little boxes in the cork. And he thought, what's those little boxes doing there? And of course, a cork is not alive. It's been taken from the bark of a tree and it's dead. So there's nothing in the cell, but the cell walls remain. And so he started looking at other things that were alive. And everywhere he looked, he saw little boxes in living things. And being that he was a religious guy, he did a lot of his work actually from a monastery. He was not himself a monk, but he had an office in a monastery. It reminded him of monks' bedrooms. Small, little boxes with not much in them, he thought, right? And so he called them cells because that's what monks called their bedrooms. So it's named after monk bedrooms. That's why cells are called what they are. So that was his thing. He named the cell. He was the first person to describe them. But he was involved in so many other things. He didn't do much cell biology. He just said, hey, look, there's these little boxes. Okay. And then a Dutch guy, go Dutch, von Leeuwenhoek. And von Leeuwenhoek had a really, really weird life. If you wind up studying him, he failed at most things that he tried to do. And then he fell into biology where he finally found success and did good things. But his early life is a story of what I'm not good at. And so, interesting guy. He studied cells, and he got all excited about cells, and he did a lot of the description of early cell biology, the parts of a cell that you can see easily with a not too high power microscope von Leeuwenhoek described. Okay, so the nucleus, and endoplasmic reticula, and some of the chloroplasts, things like that. He did a lot of that work. And then, a little while later, two Germans, Schleiden and Schwann, were working independently. Schleiden was a plant guy, and Schwann is an animal guy. And they were both doing cell biology work with their various kinds of creatures. And around the same time, came to the conclusion that, according to Schleiden, all animals were made of cells. He didn't find anything in any animals that didn't have cells. And Schwann came to the same conclusion about plants. I'm sorry, other way around. Schleiden animals, Schleiden plants, Schwann animals. And so they worked together, and they together came up with the statement that all living things are made of cells. That's their idea. And you can keep them straight because Schwann kind of looks like a swan, and he was the animal guy, right? And Schleiden kind of sounds like sliding. And I always picture going ice blocking. I don't know if you've ever been ice blocking, but you sit on a block of ice and you go sliding down a grassy hill, and it goes really fast. So I imagine sliding down a grassy hill and the grass like plants. So that's how I keep those guys straight. And those are some of the principal scientists involved in the cell theory. So those are the scientists that came up with the cell theory. What is the cell theory? The cell theory is 3 sentences. that the cell is the basic unit of all living things, that if you find something that is alive, it has cells, okay? And nothing simpler than one cell is alive. Now, again, there are some biologists that are trying to change that and say that viruses are alive, but the majority of science is not on board with that yet. So the cell is the basic unit of all living things. Cells perform all the functions of living things. That's what makes them more cool than a virus, is they do all the things that living things are supposed to do. If you remember from chapter 1, living things are supposed to grow and develop. Living things are supposed to reproduce. They're supposed to take in food molecules and assimilate them into their body. They're supposed to be irritable, respond to their environment. They're supposed to maintain homeostasis. They're supposed to move in some way. All those things that are true of life are true of cells. Cells do all that. And new cells come from the reproduction of existing cells. That statement is very important, not only for us as theists, who believe that God created the first cell and cells come from other cells and you don't have cells going poof in the corner. But it's very important as well in the story of science. The story of spontaneous generation, the idea of spontaneous generation, that if you have the right environment, cells come to be, used to be believed. And it was largely the work of Louis Pasteur who destroyed that idea and demonstrated that cells only come from pre-existing cells. So cells are reproduced from something else. Oftentimes, very quickly, we're going to do a bacteria lab later on, where we're going to start with just a couple of individual cells of bacteria, and they will, in a short order of time, become hundreds of thousands of bacteria in a colony, and that's it's only going to take 24 hours or so. So sometimes they reproduce very quickly, sometimes a little slower. So some pictures. These are some bacteria cells. These are very simple. There's not a lot to them. And so when we start looking at bacteria under a microscope, you're going to see very little more than just that, a colored, if it's been stained, little sausage-like thing in your slide. And the structures in there are very simple. There are some bacteria that are a little more complicated, that have cilia, and they move a little bit more aggressively through their environment. And as you get to more and more complicated forms of cells, some of them start to look rather intricate. This is still bacteria. This is a flagellated bacteria. It's got a little tail and it swims around. And there's more structures involved in that. There's actually a whole book written on this part of the flagella that is arguing for the existence of God based on how complicated that is. It's really cool. It's called Darwin's Black Box by Michael Bailey. Very cool book. This is a protist, and so this is a single-celled life form that's more complicated than a bacteria. It is swimming around photosynthesizing, so part plant because it's photosynthetic, but sort of animal-y because it moves around. And these cells are quite fun to study. More pictures. There you go. This is an amoeba, which is a very, very complicated cell. Still a single-cell organism, but my goodness, the amount of chemistry that goes on inside these things to make them happen. This one looks like a hand with an eyeball in the middle of it, but they change shape all the time. They like pour themselves through their environment. It's interesting. We'll get to them in a little while. This is another single-celled organism, and he's swimming around and scooping things up as he swims past them and eating whatever comes in his way. This is a colony of algal cells called volvox, and each of these clusters of cells, this is not a cell, this is a dense region of algae cells, and then they secrete this like jelly sort of bubble that they all stick to. That's really interesting. So that's a colony of algae cells. This looks like a smile, but it's actually just 3 different diatoms that were arranged in the right way to take that picture. These are 3 different single-celled organisms that somebody stuck in the right place to be able to take that smile face picture. These are algae organisms as well, growing under the surface of the water, and these are colonial. And so each cell of that plant is not actually the same organism as its neighbor. They're just all living together and they look like the plant. But that's a colony of algae as well. This is a colony of brown algae. This is giant kelp, the fastest growing organism in the world. It grows up to 2 feet a day. It's crazy. And then, of course, the sheep's head. This is an animal. It's my favorite fish to eat. I love shooting aluminum rods through the heads of those fish. Yeah, yummy, yummy in my tummy. So those are, and then, of course, some things are very, very large. California redwoods, if you've ever seen them, the largest organisms currently alive on the planet. So cells are part of all of those things. All living things are made of cells. There are 2 basic kinds of cells. There are cells that have a nucleus and cells that don't have a nucleus. So anything that does not have a nucleus falls into one of 2 groups of bacteria, either what they call archaebacteria or eubacteria. And those are very simple things that live out in the wild and in your bodies. Bacteria are in the group of cells called prokaryotic. Karyote means kernel, seed, or nucleus. It's the little thing in the middle. That's what that word means, karyote. So prokaryote means before the nucleus. Now this, red flag should go up in your brain when you see that. This is part of the evolutionary bias of biology. We still have to use the word prokaryotic because that's what they're called, but be aware that when you say prokaryotic, what you mean is before the nucleus. And so what evolutionary biologists mean is these are the cells that there were before a nucleus evolved. Okay? Failure, but we still use the vocabulary word. Okay? Prokaryotic means bacteria, no nucleus. And then eukaryotic, eu means true or good. And so this has a true nucleus. Okay? Eukaryotic cells are anything fungal, protist, algae, plants, animals, you, your dog, your cat, your mom. Okay? Cells that have a nucleus. And so eukaryotic cells, most forms of life are eukaryotic. Bacteria are prokaryotic. And there's just a diagram there to show you. A eukaryotic cell has a nucleus. A prokaryotic cell does not. They have what they call a nucleoid region, which just means a little puddle of DNA in the middle of the bacteria. There's no membrane surrounding that, making it not the rest of the cell. Okay, we already talked in the chemistry part about diffusion and things of that nature. All cells have a cellular membrane. All cells have something around them that defines what they are and what their environment is. It divides them, their juices from the environment, all the juices around them, right? And that membrane is a lipid bilayer. What does that mean? Well, lipids we covered in the last chapter. These are fat molecules, okay? And bilayer, what does that mean? 2 layers, yeah. That's a double baggie of fat, okay? If you took one Ziploc baggie made of fat and stuck it inside another Ziploc baggie made of fat, you would have a lipid bilayer, right? It's 2 layers of fat molecules that surround a cell. And it's this dual layer that controls what comes in and goes out of the cell. It's an amazing thing that, that God has put together to say, let's keep the insides in and the outsides out, and let's build it in such a way that the stuff that needs to come and go can, but let's keep the stuff that shouldn't come in out. It's incredible the amount of design that goes into this 2 layers of fat that goes around the cell. Animals and lots of protists and fungi and things like that just have a cell membrane. Other things like plants and some bacteria have what's called a cell wall, which is made of cellulose. Cellulose, remember from the last chapter also, cellulose is a whole lot of sugar molecules stuck together. So the cell membrane is fatty, the cell wall is sugary. And the cell wall is made up of cellulose, it's rigid, it's hard, and it's what gives a plant cell its shape. It's why you can build things out of dead plants, right? You can take trees and kill them and then cut them up and build houses out of them. If you tried to do that with an animal, it wouldn't work, right? Yeah. Why doesn't your house stand up? I don't know. All the dead monkeys I nailed together, it doesn't stand up, right? Dead cats, kind of rigid. Anyway, no, animals you can't use. You can't use dead animals for structure. You can use dead plants for structure, and the reason is because their cell walls are very hard, and after the inside soft part of the cell is gone, the cell wall remains and is hard and rigid. We call it wood, right? And so that's what allows us to build things out of dead plants, because they have cell walls. Animal cells, no. Okay? There are lots and lots of organelles in a cell. And I'm not going to give you all of them in this lecture because, oh dear, there's a lot of them. And also because you have already had this in life science, and because you can read and you have a textbook. But let's hit some of the really important ones. Okay? A nucleus is the thing within the cell that contains and copies the DNA. So DNA is uber important. If a cell loses its DNA, the cell will die. Because the DNA tells it how to be alive and what to do and what to make and how to repair itself and all that. That's all in the DNA. So in most cells, the eukaryotic cells that have a nucleus, there is this envelope within the cell that keeps the DNA there. It's like a big filing cabinet. It organizes the DNA. It's also where the DNA gets copied. So when the cell's about to divide, the DNA is copied inside the nucleus before the cell divides. So it's the DNA repository. And then there's the endoplasmic reticulum. That's a really long word. Really, really, really long word. Reticulae has to do with being banded. If you study zoology, there's reticulated pythons, there's reticulated sea stars, there's even reticulated lemurs, and it has to do with being striped or banded. And so a reticulum is a thing that looks banded when you look at it, right? And if you get one in your microscope and you focus it correctly to see the endoplasmic reticula, it's this. It looks kind of like spaghetti, and it's all piled up on top of itself, so it winds up looking sort of striped. Endoplasmic. Endo means inside. Plasmic means liquid. So it's inside the juice of the cell, and it's this banded thing. That's what the name means. Inside the juice, banded thing. And it's a membrane that transports materials throughout the cell. So when the nucleus makes copies of DNA and sends out these copies to be produced in a protein, the endoplasmic reticulum usually is what the cell uses to get it from point A to point B. Think of it maybe as a mail system. It's UPS within the cell. It takes something from one place and moves it along these membranes to where it needs to go. Okay? Another really important one are ribosomes. Ribosomes are the little pieces of the cell that make the proteins. They come in two parts. You'll learn more about them later. But for right now, it's the part of the cell that makes proteins. They're the protein factories. Okay? And they can either float free in the cytoplasm, or they can be on the endoplasmic reticulum. Some little bird wants it. It's funny. They can be on the endoplasmic reticulum. If they're on the endoplasmic reticulum, then it looks kind of like those sour ropes that Leah was eating a moment ago, where the ribbon of the endoplasmic reticulum is all speckled and covered with these little pebbles on it. And those are the ribosomes. And so the ribosomes on that look rough, and so it's called rough endoplasmic reticulum if it has ribosomes on it. Okay? Ribosomes make proteins. And I think I've got just one more slide of parts. Yes. This is the end of parts. So when a ribosome makes a protein, the Golgi body named after the Italian dude who discovered them. The Golgi body finishes them and gets them ready to excrete. So we talked about that proteins are not just a string of amino acids. That's the first level of organization, is the string of amino acids. But they have to be folded, and often folded proteins are woven together with other proteins for third and fourth levels of complexity. And it's the Golgi body that takes these strings of amino acids, the polypeptide chains, and folds them and weaves them and makes them what they're supposed to be so that they can be useful to the cell. So if the ribosome is like like a mass bakery, the Golgi body is the packaging plant that puts all of the little ding-dongs and plastic wrappers and boxes them up and sends them off to Times so you can buy it, right? Golgi body finishes it, okay? Mitochondria, very, very important. Mitochondria burn sugar to make ATP for the cell in the form of, make energy for the cell in the form of ATP. We're going to spend a whole week on what this guy does. I think next week we're going to spend a week on what he does. And so that's going to be something that you become very good friends with, is the breakdown of sugar in the mitochondria. The lysosomes are like stomachs in the cell, little sacs full of digestive enzymes. If a cell is a kind of cell that eats things, where foreign particles come inside the cell to be broken down as food, if that's something the cell does, then it'll have lysosomes, these little bags of digestive enzymes. And it'll take the food molecule and it'll wrap the food molecule in a membrane to make a vacuole out of it, a bag of something. And then it'll take the lysosome and a vacuole and merge them together so that the lysosome dumps its digestive enzymes all over the food and breaks it apart. Okay. So lysosomes, lysis means to break or split. Some is one of the many words that could mean a bag or a container. So a lysosome is a container that breaks things down. It contains digestive enzymes. And then a vacuole means a hole, because initially when people were studying cells, they thought that these were just empty spaces in the cell. But in fact, they are also surrounded by membranes, and a vacuole is not empty. It contains something. Usually contains water. It's where the cell will store water for use later on. Or it can contain food substances. There are some other vacuoles that contain other things. Usually food molecules or water. And so these are some of the basic cell structures that are out there. Your text will contain more of them, and I would encourage you to get to know them as you study your text for menu assignments this week. But these are the main cellular structures that we're going to need you to have in your brain as we move forward. What questions do you have? Do you remember these words from life science?
Yes.
Good, that makes me happy. Okay. If you have any questions, let me know. I will come around and stand for your behavior today. You guys were all great. Thanks very much.